In interphase, the DNA in a cell starts in the form of chromatin. Once the DNA in the cell has replicated, the cell is ready to divide. There are now 2 copies of all genetic information in the nucleus of the cell. The DNA winds up into chromosomes and becomes visible. The spindle apparatus begins to form and moves towards the middle. In metaphase, the membrane surrounding the nucleus breaks down and the spindle apparatus extends fibers to attach to the center of the chromosomes. As the spindle fibers tighten, the chromosomes line up in the middle of the cell. In anaphase, the spindle apparatus pulls the spindle fibers together, causing each chromosome to pull away and separate to opposite sides of the cell. In telophase, the chromosomes have separated and the cell will start to form a nucleus to protect the DNA. The spindle and spindle fibers release the chromatids and begin to be broken down by the cell. The cell begins to split its cell membrane and cytoplasm in half to begin the formation of 2 cells. In cytokinesis, the cell has separated and formed 2 completely new cells. The chromatids unwind back into chromatin, so the DNA can be used to create proteins. Each cell goes back into interphase and starts replicating the DNA to prepare for another round of mitosis. When the chromosomes split in anaphase, a mutation can occur when ribosomes translate a codon. Thus, a mutation is created in 2 cells when they divide in telophase. These mutated cells will continue to go through mitosis and multiply, causing the non-functional neurofibromin genes to spread in the body.
Central dogma is the process of transcription and translation. When transcription occurs in the nucleus, RNA is replicated and then read by the RNA polymerase, producing mRNA. The newly created mRNA moves to the cytoplasm and occurs translation. During translation, ribosomes read the mRNA and transfer it to assemble an amino acid, forming a polypeptide chain, and then finally creating a protein.
During the central dogma process that revolves around the genetic disorder of neurofibromatosis type 1, the transcription and translation happen typically with some complications. In Kai's case, he experiences a frameshift mutation. It causes the deletion of a CG sequence and an insertion of an ATAC sequence, changing multiple amino acids, thus resulting in a dysfunctional neurofibromin. Messing up the sequence of 3 amino acids causes the mutation that can later become visible physically or show up in the genetics of a person and their offspring.